Hi class, now I'm going to teach you the chapter titled Stem Cells. So before I go into stem cells like I did with earlier chapters, I would like to give you some basic concepts. But before even I go into basic concepts, I just want to go over something very important that one should realize that our body, a human body, is the fundamental unit of life is a cell. These identical cells, they come and form tissue, and an identical tissue, they make an organ, and then one organ can work as a system to support our body. So this is a hierarchy in our body, right? So there is a defined way our body is organized. It is not haphazard. So if you take an example, you could consider a cell in a heart, right? And the identical cell, they form heart tissue, right? And then it becomes a tissue becomes an organ, and then heart controls the circulatory system. And what is the function? It carries blood. Through blood, it delivers oxygen. It takes off the carbon dioxide, the impure thing, and then that's we exhale through lungs. So what is happening is there's a system, and this circulatory system, this collaborates with respiratory system. Respiratory system, the master organ is lungs. Lungs is the place where oxygen and carbon dioxide are exchanged. So our body is an extraordinary model for teamwork, collaboration, and it's just magical. But our body runs into diseases, and diseases have been occurring since many years, and it's becoming more complicated. So we need to have alternative approach to treatment. We have to bring more and more technology and innovative ideas to generate new capabilities of therapeutic design, right? And that, of course, we are doing. So so this is the hierarchy. So what happens that I'll relate this to a problem. Let's say let's say there is an organ failure. Let's say a kidney has failed in a person, which is a very traumatic experience. And what is happening that the current therapeutic modalities has been for these organ failure is, let's say, kidney transplant. Okay? But this is not easy. Why this is not easy? This is not easy because first, there is a long, right? There's a long queue. And then what is happening is that even if you find a donor, the problem is still there because it may not match. Tissue may not match. Matching may not be perfect. So if matching is not perfect, there will be acute or chronic rejection, depends, right? So what is an alternative to this to treat organ failure? Also, there are many individuals who run into accidents or people who go for war. They can lose their organ parts. How to replace that, right? And so this is a important issue, important area of research to find solution. So one of the strategy is using stem cells. Stem cells. Now, what are stem cells? To describe it in a very simple words, it is mother of all cells, which has a potential to give rise to an organ organ or an individual. To an individual. For example, why do I said individual? I said individual because let's say in case of in normal situation when fertilization occurs between male and female gametes and there is zygote, zygote has a program inside. Zygote is a single cell and it has a program that can lead to the development of whole individual. Right? So zygote has that capability to. So so let's consider a situation where there is an organ failure, right? There is an organ failure in an individual, and there is no chance. There are no chances to find a good match. The chances are very bleak, right? Very bleak to find. Very bleak chances to find a match donor, right? So what scientists are trying to find is to find a potential cell that can be grown, and it can take the shape of the diseased organ. So essentially a cell that can be educated to develop into a disease organ. It can substitute the disease organ such that a person becomes normal. Okay, so this is the basic concept of this chapter, which is called as stem cells, right? So now our body is very planned, right? Human body or any organism is very planned. It's very sophisticated, right? And I remember when I was teaching cell division, I told you that every cell every individual, whether it's a cell or a bacteria or a plant cell, they want to grow. Growth, development, and differentiation is an essence of life, of a living being, rather, right? So growth, development. So for example, let's say zygote, right? So zygote is a cell after fertilization. It has a capability to develop total organism. This could be human body or animal body, total organism. So this potential is called as totipotency. So when a cell has the ability to develop into a whole organism, that cell is called as a totipotent cell. So what does potency means? Potency means that the potential to grow. Toti means total. So zygote is a totipotent cell. Now, what happens that after zygote is formed, the single cell, the cells starts dividing, right? And when the cells start dividing, they form 3 layers of cells. Top layer, then the middle layer, and then the inner layer, right? So this zygote, it develops into 3 layers, right? These 3 layers of cells. Now each layer has its own name depending upon the location. So for example, the outer layer is called as ectoderm. Okay. The middle layer layer is called as mesoderm. Mesoderm. So this is the middle layer, and the inner layer is called as endoderm. Now, why I'm sharing this information with you? I'm sharing this information with you because these three layers, 1, 2, 3, these three layers can lead to the development of different kinds of tissue. So different layers can give rise to different cells with different cell fate. So for example, ectoderm, the outer layer can form skin and nervous system. This layer is very important because it's very good for protection, right? The middle layer can give rise to muscle cells and bone marrow. Why bone marrow? Because bone marrow are very responsible for giving blood cells that we'll study soon. Next slide. And then the inner layer will give rise to all the systems like circulatory system, alimentary canal, the digestive canal, the kidney, respiratory, excretory system. So these three layers are very serious thing, right? They have different cell fate regarding to what system they give rise to. So the capacity of this three layers, the capacity of these three layers, capacity of these three layers is called as pluri. So what we have covered up till now, totipotency in zygote, and when zygote forms three different layers, ecto, meso, and endo, they give rise to different system that is called as pluripotency, right? Now, let's go into bone marrow cells. Now, bone marrow cells are those bone marrow is those tissue that give rise to our blood cells. So blood cells can be of many kinds. So what you have to appreciate that the mother cell is bone marrow cells, but the bone marrow cell has capability. Bone marrow cells have capability to give rise to many cells. So bone marrow has capability to give rise to many cells. For example, one of the categories could be RBC, yeah, the red blood cells. Right? Second category could be WBC, which are white blood cells. The third category would be platelets. So all these three cells, different kinds of cells, they're not same. They're different kinds. Why they're different kind? Because they have different functions. What RBC does? RBC is involved. They carry hemoglobin, and they are involved in oxygen and carbon dioxide exchange. Right? The hemoglobin, as I mentioned earlier, carbon dioxide exchange. WBC, it is involved in defense mechanism, fighting infection. In other words, they're involved in immunity, right? And third kind of cells, which I mentioned to you as platelets, they are involved in clotting. If our blood, so we get many micro injuries and injuries in a daytime, and if our blood doesn't clot at the site of injury, then the blood will keep flowing. So platelets are involved in. So, but what I want you to emphasize here, what I want you to understand here, the significance that all these cells, right? All these cells, they are emerging from bone marrow, right? So bone marrow have certainly tremendous capacity, potential. So the potential of bone marrow to develop into these cells, which they're unique by their function, structure, and mainly structure and function. So the potential of this bone marrow is called as multipotent. Multipotent. So what we talked about, totipotent, zygote, pluripotent, on the three germ layers, ecto, endo, and meso. And then bone marrow is an example of a multipotent that give rise to RBC, WBC, and platelets. Okay. Now let's consider only the WBCs. Right? So WBCs, as I mentioned to you, are the important cells involved in defense mechanism in protecting us from pollution, from viruses, bacteria, dust, many things. So WBC are, if you will be surprised to know, WBC is made up of many kinds of cells. So for example, WBC could be of lymphocytes, right? WBC could also be made up of eosinophils, participates in allergy, right? Then basophils, that participates in let's say an infection from worm, right? In many countries, tropical countries, we get infected with stomach worms that creates a lot of digestive problems. And there are more cells like neutrophils that also helps in attacking the bacteria. But let's focus on lymphocytes. So lymphocytes, it has two branches, number 1 and number 2. Number 1 is T cells, and number 2 is B cells. So what do you see here? That to start with, there is bone marrow, then bone marrow gives rise to WBC, right? WBC has 3 or many components, but for this class, let's focus on lymphocytes, and lymphocyte give rise to two population of cells. Such lineage, such potential of lymphocyte is called as oligopotency. Right. So what we talked again, totipotent, pluripotent, multipotent, and oligopotency. The last is called as unipotent. So there are some cells in our body that lose their ability to grow as we age, and as such they grow, they divide very slowly, and they're very precious organs. For example, liver, kidney. So they divide very slowly, and if there is if there's an injury, then it's very difficult to regenerate these cells, right? So, for example, liver is made up of hepatocytes. So, if there is liver cirrhosis, chronic jaundice, hepatitis, then the hepatocytes will be damaged. It will be hard to replace. So some people are recommended for liver transplant because they cannot grow. Same with brain. If somebody has a brain stroke, and so let's say nerve cells, right, or neuronal cells damaged, it's very hard to. So some people who get third-degree burn or higher, serious burn, their nerve also is destroyed. So it's very hard to get that functional. Same with kidney. So these cells, they divide very slowly as compared to skin cells, as compared to our hair follicle. So these are called as unipotent cells. So this, what I discussed with you, the potential of various cells in our body, how they grow, how they mature, and what are their level of growth. So now I would like to shift gear into, so there are stem cells are those cells which can give rise to a tissue or an organ. So some of this type of stem cells are human embryonic stem cells. Then there are adult stem cells. Then there are, let's say, cord blood cells. And most of the recent research is focusing on a special cell called as induced pluripotent cells. So I have seen a couple of students presenting on stem cells. So I hope it will make more sense to them. So human embryonic stem cells, which is also otherwise called as HESC cells. So human embryonic stem cells have been controversial because to develop human embryonic stem cells, you have to deal with human embryo, and there is a lot of debate going on. So what are human embryonic stem cells? So after 5 days of growth, right? 5 days of growth, the human embryo takes a shape, and this shape is called as blastocyst. Okay? So this is fifth day of embryogenesis. Fifth day of embryogenesis. Now, on this 5th day of embryogenesis, what happens that there is a special cells that are formed inside this. Special cells. Okay? Special cells that are formed. And they don't occupy, but they are in the corner. Right? These special cells are called as inner mass cells. Right? They're called as inner mass cells. These inner mass cells are the human embryonic stem cells. Right? Now, if scientists, they want to work with human embryonic stem cells, they have to reach out to the human embryo, and then that's why it could be controversial, a topic of debate, whether to to use human research, human embryonic stem cells for research. But it has huge potential because these human embryonic stem cells are naive cells. They are naive and they could be developed to replace any diseased organ. Right? So this is a little bit about human embryonic stem cells, where they are found, and their potential. Right? Now the second type of stem cell is adult stem cells. So when the research on human embryonic stem cells were not encouraged, then scientists had to develop alternate system, and they started working on adult stem cells. So remember, human embryonic stem cells, they're found in embryo, which is 5 days old. They are very naive. Cells have not been exposed to environment. They have not been exposed to pollution. They are extremely easy to handle, but adult stem cells are derived from adult organs, right? For example, let's say if we want to work with, to develop nerve cells, then we have to work with nervous system, right? So we have to, let's take a situation where there is a brain stroke and brain tissue and nerves have been damaged because of stroke, and they have to be replaced. So, and this is in an adult person, and there is no availability of human embryonic stem cells. Then stem cells, adult stem cells could be derived from adult brain or nerve tissue, and that can replace the part of the brain that has been affected due to brain stroke. But adult stem cells are extremely rigid. Why rigid? Because they have been exposed to so much of environment, pollution, and so it's difficult to educate them to become into a new organ. So it's very, very difficult. It's easier to work with human embryonic stem cells than the adult stem cells. Then now we are learning about cord blood cells to be potential for stem cells, but we still don't know exactly whether it is a good idea and it has a therapeutic impact. So these are newly developed technologies that still needs to be tested, but it's going in the right direction, right? So the last one, which is the most interesting, is called as induced pluripotent stem cells. So here the concept is that let's say when we pass urine or when we pass our food through our intestine, a lot of cells come out, right? So let's say a cell from urine, right? These cells from urine can be taken out, and these cells can be in these urinary cells, which is of no use to us. It is being passed down, so we can collect those urine cells, and then we can put 4 genes into these urine cells. For example, c-MYC, NANOG, then KLF 2 or 4, and then OCT2, right? And so these are special proteins which are called as transcription factors. So these transcription factors, if they are injected into urine cells, right, and this urine cell is given good right environment, so we can develop this. So what is the concept here? Induced. Urine cells can be induced with this 4 gene to be grown in the lab conditions to provide the right environment so that this urine cells could grow into heart cells, brain cells, kidney cells. So this is induced pluripotent cells. So stem cells. So with this, we are almost finished to stem cells. This is blastocyst. This is inner cell mass, right? This is blastocell 5-day stage. Right? And they can, so this is So this slide will show you various terminologies: embryonic stem cells, pluripotent stem cells, adult stem cells. What are the difference between embryonic and adult stem cells? And their capacity to grow, their capacity to differentiate, their capacity to be comfortable in the lab situation. So with this, I finish the chapter on stem cells. Thank you.